Hi everyone, it's Mr. Stratton. Today I want to talk to you about carbohydrates. As with all of our biomolecules, the thing we're most interested in is the relationship between structure and function. In this case, primarily we're looking at energy and actual body structure. So that's the functions of carbohydrates, so short-term energy storage and structure. And then the monomer would be a monosaccharide, mono meaning one, saccharide meaning sugar, and the polymer is a polysaccharide, many sugars. The general structure, the stereotypical structure, is this ring structure, although they can occur both as rings and as in linear format. But being a lazy biologist, I'm pretty much going to draw my carbohydrates, my monosaccharides, as little hexagons. However, not all of them are hexagons. In the case of fructose, it's actually a pentagon. But the general formula for a monosaccharide is CH2O to the X. So in this case, if you're talking about glucose, that X would be 6. It'd be C6H12O6. Probably remember that from freshman biology. Polysaccharides are made up of many monosaccharides. So that can be a disaccharide, meaning 2, a trisaccharide, meaning 3, or a polysaccharide, meaning many. Again, you're talking about things like sucrose, which is a disaccharide. That's table sugar. And maltose, which is malt sugar. The polysaccharides we're interested in, cellulose and amylose, which we'll get into a little more detail, but they are many of those monosaccharides in long chains, hundreds of them in many cases. You may notice that the names of sugars they all end in ose, right? Glucose, fructose, sucrose, cellulose. So I want you to figure out what's the name of this molecule. Okay, the way they're put together is in dehydration synthesis. So again, we're going to remove a water molecule. The hydroxyl group is going to react with a hydrogen off of another monosaccharide, and it's going to form a disaccharide in this case. That linkage between the two monosaccharides has a special name. It's called a glycosidic linkage. They're also degraded through hydrolysis. We're going to throw that water back in there, the hydroxyl group and the hydrogen, separating out those two monosaccharides from each other. Okay, function-wise, short-term energy storage. So that would be long chains of monosaccharides, so the polysaccharide, that you can then hydrolyze and break off those individual monosaccharides, throw them into the mitochondria, and produce ATP. The two main ones we're going to talk about, amylose is plant starch. So plants photosynthesize and produce sugar, produce glucose, which is ultimately then going to be either used or it's going to be stored. So a potato, it's made up of starch, amylose. Glycogen, on the other hand, is found in animals. So animal starch, glycogen, is made up of these long branch chains. Now, it's not that different from amylose. Same general function. Again, you're going to put all these glucoses together in your liver and in your muscles as well, and store that sugar for later use. So if you eat a potato, you eat a French fry, you're going to digest that amylose using the enzyme amylase, which is in your saliva and your intestines, and then you're going to either use it in your mitochondria or store it in your muscles or your liver as glycogen. Okay, now the name amylase, ASE on the end, that suffix, that indicates the enzyme. So amylose is broken down by amylase. All right, functions of structural polysaccharides. These are ones that actually form up the body structure of organisms, and primarily we're talking plants here, right? We don't have structural polysaccharides in our body, but you'll notice that these are long chains of glucose that are cross-linked. That's forming some nice, tough fibers called cellulose that make up wood. Okay, so the difference between starch and cellulose is the linkages. So the chains are alternating alpha and beta in cellulose, whereas they were just alpha glucose molecules in amylose. Now you probably can't see the difference just by looking at this molecule, but if I put them side by side, you'll notice that in amylose, which is on the bottom, it has all of its glycosidic linkages on the same side. They all face the same direction, and our enzyme can get in there and chop that up no problem. In fact, it starts in your mouth. Cellulose, we do not have the enzyme cellulase, which is required to break down those bonds because they alternate up and down and up and down. Now, what has that? Well, you're talking about things like fungi, so mushrooms, they break down dead trees, stuff like that. Also, termites don't, but they have a symbiotic protist in their gut that does. Now, just an interesting side note, this is what fiber looks like. That's what we call cellulose. But that bond, well, we don't have the enzyme to break that down. And in fact, the enzyme to break that down didn't evolve until many, many, many years after cellulose first evolved. And so the very first organisms that had cellulose, when they died, there was nothing that could break them down. So that the trees, which, by the way, trees have been around for less time than sharks have. They've only been around for 350 million years or so. So 350, 300 million years ago, there was nothing that could break down cellulose. The enzyme had not evolved yet. And so those trees, when they fell down, they did not decompose. There was nothing that could break them down. And so what they did was they formed coal. And our coal deposits from the Carboniferous period, they truly are a non-renewable resource. Coal will never be made again, simply because nowadays if a tree falls over, there are fungi jumping on that thing and breaking it down with their cellulase enzyme. So again, we call this fiber. You can tell that this is pretty strong. If you want to see this yourself, take a piece of paper and tear it and hold the torn edge up to light, or even better, if you have a microscope, take a look and you can actually see the cellulose fibers within the paper. The last structural polysaccharide I want to talk about is chitin. Yeah, I'm not chitting you. This is actually pronounced chitin, and that is found in fungal cell walls and insect exoskeletons. Now, those seem like two very dissimilar places, and it actually gives us a clue about evolution, because those two organisms are more closely related, and fungi are actually more closely related to animals than plants. It makes sense because they have more similar metabolism too. They don't photosynthesize. But it's got weird little nitrogen side chains off of it. You don't have to memorize the structure, but it is just sort of an interesting evolutionary side note. So what I want you guys to do now is explain how the structure of starch and cellulose are related to their functions, and explain why it is we're able to digest starch but not able to digest cellulose. Thanks for watching.